So let's start with one component at a time. The first one is the collagen, which is the major structural protein of most of the connective tissues. About 25 to 30% of the total weight of protein in the body is collagen. It serves to hold together the cells in tissues. It is the major fibrous element of tissues like in our bones, in our teeth, tendons, cartilage and blood vessels. At least 28 distinct types of collagen made up of over 30 distinct polypeptide chains, each encoded by separate genes, have been identified in human tissues. The most abundant ones are the type 1, type 2, type 3 and type 4 collagen, and the rest of them, as you can see in the chart, is mentioned here. So, the type 1 collagen is the most abundant form seen in the connective tissues in almost all regions of our body. The type 2 is mainly seen in cartilage and vitreous humor. Type 3 is seen in skin, lung and vascular tissues, that which require more elasticity, and type 4 is seen in basement membranes. Functions of collagen. The first one is to give support to organs. Second, to provide alignment of cells so that cell anchoring is possible. This in turn helps in proliferation and differentiation of cells. And lastly, in blood vessels, if collagen is exposed, platelets adhere and thrombus formation or clot formation is initiated. Now let's discuss the structure of collagen. Collagen has a triple helical structure, each made up of a polypeptide chain. Now each polypeptide chain of collagen, it has about 1000 amino acid residues, and every third amino acid is a glycine. The repetitive amino acid sequence may be represented as glycine XY, where X and Y are other amino acids. So most commonly they are proline and hydroxyproline. Moreover, 4-hydroxyproline and 5-hydroxylysine are also found large proportions in collagen. Proline and hydroxyproline confer rigidity on the collagen molecule. Next, let's learn the synthesis of collagen. Collagen is synthesized by fibroblast intracellularly as a large precursor called procollagen. After the synthesis intracellularly, it is then secreted outside the cell. The extracellular procollagen is cleaved by specific peptidases to form tropocollagen. Next is post-translational modifications. The hydroxylation of proline and lysine residues of collagen is a post-translational modification taking place intracellularly. Now, prolyl hydroxylase and lysyl hydroxylase enzymes also contain ferrous ion in its active state, and it requires a reducing agent like ascorbic acid to preserve the ion in the reduced ferrous state. So, vitamin C deficiency can lead to poor hydroxylation. It is the major biological defect in scurvy. So, next is extracellular maturation of collagen. So, as we discussed earlier, procollagen molecules are secreted out of the cell. Now, outside the cell, this procollagen, it is cleaved by specific peptidases. Finally, covalent crosslinks are formed. If there is a deficiency of this peptidase, it can lead to a condition called dermatopraxis, where the skin is prone to be torn easily. Triple-stranded helix. The collagen is a rod-like structure. Each of the 3 polypeptide chains, it is held in a helical conformation by winding around each other. Thus, a suprahelical cable is made. The 3 strands are hydrogen bonded to each other, and the amino acid named glycine, because of its small size, fits into this crowded interior of the collagen triple helix. The next is quarter staggered arrangement. The tropocollagen molecules are arranged in a quarter staggered array to form collagen fibers. Now, this structure repeats after each row. Thus, the collagen fiber has a triple stranded quarter staggered arrangement which helps in mineralization. Crosslinks in collagen fibers. The collagen fibers are strengthened by covalent crosslinks between lysine and hydroxylysine residues. Thus, crosslinks are formed by lysyl oxidase, which converts these amino acids to aldehydes. The aldehyde derivatives of these lysine residues can form an aldol condensation. Such aldol crosslinks are formed near the amino terminal of the chains. These are called the aldol crosslinks. So, lysyl oxidase is a copper containing enzyme, the copper ion being located at the active site. In copper deficiency, the copper links of the collagen are reduced. Abnormalities in collagen. So, the first condition here is osteogenesis imperfecta, inherited as a dominant trait. It is the result of a mutation which results in the replacement of this single glycine residue by cysteine in the type 1 collagen. This change disrupts the triple helix structure near the carboxy terminus. Hence, the polypeptide becomes excessively glycosylated and hydroxylated, and this results in brittle bones leading to multiple fractures and skeletal deformities. The second condition is Ehlers-Danlos syndrome. This results from the defective type 3 collagen formation due to defective enzymes lysyl oxidase or lysyl hydroxylase. It is characterized by weakening of collagen, loose skin, hypermobile and lax joints, as you can see in the image. So, the third condition is Marfan syndrome. It is inherited as an autosomal dominant manner. Arachnodactyly, which is long digits, ectopia lentis, which is dislocation of lens, hyperextension of joints, and aortic aneurysm are the manifestations shown. This disease is produced by a defect in the gene coding for connective tissue protein named as fibrillin 1. It is a component of microfibrils which normally gives the substratum for deposition of elastin. So, fibrillin and elastin are deposited at low concentrations here. Now, the fourth condition is Menkey's disease. So, deficiency of copper results in defective function of an enzyme called lysyl oxidase and reduced cross-linking of collagen results. So, the fifth condition is the deficiency of ascorbic acid. It is characterized by the defective hydroxylation of collagen. The collagen formed here is weak, leading to fragility of blood vessels, poor wound healing, etc.